Microbiology from Institute for Medical Research, that is IMR. I will be your moderator for this afternoon. Also, I would like to acknowledge the organizer for this webinar, that is Malaysian Mycology Interest Groups under the Malaysian Society for Infectious Disease and Chemotherapy. In short, it is known as MSIDC Malaysia. This is our organizer for this webinar series. So this is a serial webinar which is open for all, including researchers, officers, lecturers, allied hairstyles, laboratory technicians, and even a student can join if you are interested in the field of medical mycology. Okay, so for today, the topic of our webinar is very interesting. It is fungal identification, learning from basic at its best. The eyes can only appreciate what the mind knows. Our speaker for today is Dr. Muhammad Fuat bin Abdul Razak. Okay, but before we start, I would like to highlight a few housekeeping messages. Okay, this, as we all know, we are in a Zoom features where everyone here, you as participants, might only see yourself as the only one in this webinar, but actually there are already about 52 participants, and this is a feature where you cannot view other participants, but you are not the only one in here. Okay, so we already have a number of crowd together with us. Okay, so throughout this session you are muted, but at the end of the presentation by our speaker, we will have a question and answer session. But throughout the presentation, feel free to drop any questions or any comments in the chat box. Okay. So we will also taking questions. You can speak out at the end, where we will allow participant to raise hand, and we will unmute the participant as well. To our audience, all of you has registered for this webinar previously, so means you are entitled to claim MMA CPD point. So you can do this by accessing our poster for this webinar and just scan the QR code, and you will be automatically led to the MMA CPD point site. Okay, now I will introduce our renowned speaker, Dr. Muhammad Fuat bin Abdul Razak. He is a bachelor degree holder from University Malaya, Kuala Lumpur. So he holds a Bachelor of Science degree in the field of microbiology. He obtained his Master's Science degree in the year 2000 from University Malaya in the field of microbiology, specializing in anaerobic bacteriology. Subsequently, Dr. Fuat graduated as Doctor of Philosophy with PhD degree holder from Glasgow Caledonian University, Scotland, United Kingdom in the year 2006 in the field of toxicology, specifically microbial metabolites. So he was previously research officer in Institute for Medical Research from the year 1993 to 2020, that is 27 years of service in total. He has in-depth experience in medical microbiology throughout his career, and he has deep knowledge in fungal identification. He was the supervisor of Medical Microbiology Laboratory in IMR from 1993 to 2019, and involved actively in identification of mold and yeast, susceptibility testing for fungi and culture, as well as identification of fungi from clinical specimens. He also a lecturer in Diploma of Medical Microbiology, that is a postgraduate diploma course offered in IMR under Semiotrop-Med. So he used to give us lectures, and now also actively giving lectures in this course for yeast identification, mold identification, as well as diagnosis of mycosis. And throughout his career, he's involved in various research projects together with with local and international collaborators with multiple publications in local as well as international journals. So without further ado, I would like to invite our speaker for today, Dr. Fuat bin Abdul Razak, to deliver his topic entitled Fungal Identification, Learning Basic at its Best. The eyes can only appreciate what the mind knows. Okay, please welcome Dr. Fuat. The floor is yours, Dr. Fuat.
Okay, Assalamualaikum warahmatullahi wabarakatuh. A very good afternoon and salam sejahtera. Thank you very much, Dr. Murnihayati, for the kind introduction. Today, I want to give a talk on the titles of Mohd identification by cellular morphology, difficulties and tips. I'll be spending maybe nearly 1.5 hours on this topic. I hope all of the participants can stay put with me until end of this presentation. By the way, this is yeah, it's interesting topic, but maybe it's not easy to understand. And because you have to visualize what I say, so I prepared a lots of photos to help you to understand my talk. Few years ago, I was invited actually to a mycology lab somewhere in Klang Valley, and I was asked to check their fungal isolation and identification. So I went through their record book, and surprisingly, I found they have identified quite a number of Coccidioides immitis. It was a surprise to me because Coccidioides immitis is not in Malaysian soil. So how could this fungus was found by them? So this is Coccidioides. It show arthroconidia and disjunctor cell are alternately ranged on hyphae. So this is the description for Coccidioides immitis. It has arthroconidia, the spore-forming. I mean, this is the reproduction structure of Coccidioides, and there are also disjunctor cell in between two arthroconidia. And they told me that they prepare sellotape mound of the fungal isolate, and then it was observed under microscope, and they have reference atlas to confirm their identification. So this is what we can see in the most mycological atlas. So in mycological atlas, there will be either drawing or photos of fungus, and there will also be there the morphological description of the genus, and maybe some atlas will also have key to species within the genus, and they will also describe a morphological description and pathogenicity information of the species in the genus. So this is what most mycological atlas will have in their writing. Okay? So I think this when they look at the atlas, they probably miss another fungus which look like very similar to Coccidioides. This fungus is called Malbranchia. See, as in Coccidioides, Malbranchia also has arthroconidia and disjunctor cell in between two arthroconidia. So looks very similar if you look at the photo. So you may think this fungus are the same fungus, okay? But if you read the descriptions in the atlas, then you will see there is at least one difference between Coccidioides and Malbranchia. So Coccidioides, the Atroconidia, are cylindrical to barrel shape. So this is a barrel shapes, okay? But in Malbranchia, the Atroconidia are cylindrical with truncate ends. So this part cylindrical can be longer than this. This is the truncate end, okay? So if you look carefully again at the photos, then you will see, you can see here, these are the barrel shapes Atroconidia. This is the cylindrical with truncate ends. So then I believe what they saw in their lab was not Coccidioides, but Malbranchia. Okay, so each genus and species of moths has key morphological characters. So if you don't learn these key morphological characters, you will depend heavily on comparing what you see under microscope with photos in the atlas. Okay, when we observe an unknown mold under microscope, we look for these key morphological characters that will lead to the identity of the mold. Okay, so I have two examples. One is this fungus. So if you see something like this under microscope, then you have to look for key characters, key morphological characters. So actually this fungus has several key characters. One is the hyphae, the dark hyphae. This dark hyphae is called the multi-axis hyphae, and it has flask-shaped conidiogenous cell. So this is the cell that produces the conidia, and this is other conidia, but the conidia is in a sticky mass form, or we call it ball of conidia. And there is also at the end of this conidiogenous cell structure, we call it collarette. And the collarette is flaring and darker than the conidiogenous cell. Okay. So another example is this one. Okay, if you look at this fungus under microscope, so what are the key characters? The key characters is the multi-axis hyphae. So see, this is the colony of the multi-axis fungus. So this fungus has conidia with one septum. So we call this one the diamo conidia. And these conidia are produced from this structure. We call this structure conidiophore, but this conidiophore is not straight, but it's sympodial or geniculate. And there are bud scars on the conidiophore. So if you have these 3 or 4 key characters, then you can identify this fungus. Okay. So identifying the key morphological characters is essential in the process of identifying mold by cellular morphology, but this is not always easy. Okay, that is the why today we have this presentation. Maybe I can share some of my experience with all the participants. Some of you probably just started learning how to identify mold. Some of you probably have done it yeah, maybe for a few years, but still think that you need to learn something on this mold identification. Okay, so that was my actually introduction to my talk. So for today's talk, the lecture outline is very simple. I have first part of my lecture, of my talk, will be on the discussing key morphological characters in modes. Because we can't identify a mode without knowing these key morphological characters. And I think there is probably not many places you can learn this. So number two, after that, I will discuss some tips in studying key morphological characters in modes. So how are you going to start studying it? Okay, where you can study these key morphological characters in modes. Okay, at least today, this is the first day to some of you learn about very important mycological terms related to key morphological characters. Okay, so basic cellular structure of filamentous fungi are hyphae, and hyphae came from conidia, and these hyphae elongate at the tips, and it form branches. Okay, a network of hyphae is called mycelium. So fungi which have only mycelium without yeast cells are called modes. Okay? So life cycles of hypomycete is like this. Okay, so once the fungus produce mycelium, so the hyphae will produce specialized cells. These specialized cells will produce conidia. So this conidia will later germinate and form hyphae again. So this is the life cycles of hypomycete. But some fungi, when grown on routine laboratory media such as seborrheic dextrose, brain heart infusion, or even on sporulation media such as potato dextrose agar, do not produce asexual conidia. So, if we study the hyphae carefully, if we observe it carefully, so it may reveal some key morphological characters, even though we don't see conidia. Because, for example, some fungus such as mushroom won't sporulate on seborrheic, won't sporulate on BHI, so we will see only hyphae. So we have to study the hyphae to see if you can identify some structure that you can make conclusion that the fungus you are looking at is a mushroom. Okay, fungal mycelium from colonies on seborrheic agar. So first of all, when you look Mohd under microscope, of course you will see hyphae. Next, you have to identify whether the hyphae is hyaline or dermatiaceous. Okay, hyaline means it has no pigment on its cell wall. So when you stain the hyphae with LPCB, it will turn to blue hyphae. But there are fungus, whether you stain it with LPCB or you look at straight away under microscope without staining it, so it looks similar. They look brown to black. The hyphae is brown to black color. Okay, so because this group of fungus produce pigment, mostly melanin on its cell wall. So, and this melanin is not stained by LPCB. So the fungus look brownish to black. So fungus with this kind of hyphae or mycelium is called dermatiaceous mold. Or if you like, you can call them black mold. Okay, but I would prefer the multi-Asians because it's not really black. It is actually brownish, dark brownish, and when it's the molds has been matured, it's probably turned to blackish. So the multi-Asians hype, it's very important. Don't jump straight away. I mean, when you look under microscope, don't jump to look for conidia, look for all these protein bodies, but determine whether the hype is the multi-Asians or not. Okay, because if you can confirm the hype is the multi-Asians, then you are looking at the multi-Asians of black molds. So there are over 100 species of the multi-Asians mold implicated in human disease. And these 100 species are in 60 genera of mold. So these are some of the common, the multi-Asians of black mold you can isolate from clinical specimen. Okay, but the problem is if you look under microscope, sometimes it's not easy to say whether this is the multi-Asians or highline. If you look here, so the hype is bluish because it was stained with LPCB. So you may think this is highline mold. But look at the conidia. The conidia are the multi-Asians. So how do you what easier way to confirm whether the fungus is the multi-Asians or not is by looking at their colony, because the multi-Asians molds show brown to black color, whether from the tops of the colony or the reverse. Okay, sometimes you see the black color at the reverse of the colony. So this is enough to confirm that the mold is the multi-Asians mold. Okay, it's very important because if you see the multi-Asians mold and after careful observation you don't see a conidia, you probably can subculture this multi-Asians mold on a better media that most multi-Asians mold was sporulate, because we normally isolate fungus from Sebrot dextrosaga. So because Sebrot dextrosaga is the most commonly used routine primary isolation media. Okay. Sterile cynocytic hype. So we move from the multi-Asian hype to sterile cynocytic hype. If you look under microscope, some mold will have crossfall in its hype. So this crossfall is called septum. Okay, fungus with septums, or you can see the septum easily, are called septate hype. Okay, there are groups of mold. In this group, it's hardly to see the septum. Not because the septums are not there, but they are probably far apart from one another, very sparsely in the hype. Okay, so these groups of mold, we call it. This hype is called cynocytic hype. Cynocytic hype, or if you read all the textbook or even modern textbook, the author probably named this hype as aseptate. But I would prefer to use cynocytic rather than aseptate, because aseptate means no septum at all. But actually this cynocytic hype They are septum. They are septum. But if you look here under this one field view of microscope, you don't even see any single septum. Okay, so cynocytic hyphae. So this observation is very important. This is how cynocytic hyphae looks like. So there is no septum here actually. And if you look under microscope and you see cynocytic hyphae, then you are probably looking at two big groups of fungus. One is zygomycete, the other one is oomycete. Okay. What's next in zygomycete? In zygomycete, the hyphal waist is about 6. I think it's not 16. It can be bigger than this, up to even 20 plus to 30 micrometers. Some zygomycete has very big, very wide hyphae. So, but even 6 to 16 is considered broad hyphae. So, zygomycete not only cynocytic but has broad hyphae. So, if you compare zygomycete hyphae to aspergillus, so I mean, most of us probably have seen aspergillus under microscope. So you probably are used to see the waist of aspergillus hyphae. So that hyphae waist is about 2 to 3 micron. So it's probably 1/3 of the waist of zygomycete. Okay. If you haven't seen aspergillus yet, probably you have seen Candida. So Candida size is about 2 to 3 micrometer wide also. Okay. So my tips, if you want to make sure you know how white hyphae look like, you probably can start learning it by looking at aspergillus hyphae. Okay, so then you know if you look a bigger, wider hyphae than that, then it's probably zygomycete. Okay, so I have a diagram here comparing aspergillus to zygomycete. So how do you differentiate zygomycete from oomycete? And because both of this group are cynocytic hyphae, so zygomycete has broader hyphae than oomycete. Oomycete hyphae is broader than aspergillus. So that's one way how we differentiate zygomycete from oomycete by looking at the waist of the hyphae. Okay. If you look at the microscope, you see cynocytic hyphae and then white hyphae too. So one way to confirm that the fungus are zygomycete by looking at the colony. So zygomycete has what we call it cottony colony. Okay, while the oomycete, if you look at the colony, it's almost glabrous. Okay, so then we are finished with oomycete and zygomycete. So if you see only broad hyphae, but you don't see any spore, then but you can still have an idea that what are you looking at is probably zygomycete. Okay, basidiomycete and mushroom. So mushroom, we have before my retirement. In IMR, isolated a lot of mushroom from clinical specimen. Okay, but none of these mushrooms sporulate on cell rod or on potato Dextrosega. So mushroom on sap, you will see sterile mycelium, meaning that there is no conidia, no spore. But you probably see this structure, we call it the clamp connection. It's a hook-like structure formed by growing helper cells. So this clamp connection, if you see it, then the fungus is Basidiomycota, or such as mushrooms. Okay. So this is the Basidiomycota, basidiomycete. Actually, basidiomycete, I think more than half of the fungus identified in the world today are in this group. Okay, it's a very big group of fungi. Okay, let's move on to hypaton arthroconidia. Okay, in many fungi, the hyphae disarticulate into arthroconidia, and it's probably the only asexual conidia seen in large number of fungi species. So these arthroconidia are seen in large number of fungi species. So we normally don't use the characteristic of arthroconidia for identification. This is how arthroconidia are formed. Okay, segmentation of pre-existing fungal hyphae. Okay, there are two types of arthroconidia. This type, arthroconidia, are formed without disjunctive cells. So this is a disjunctive cell. Actually, disjunctive cells are just an empty cell that fragment and undergoes lysis to release the arthroconidia. So Coccidioidetes, fungus that I spoke at the beginning of my talk, is probably the most famous fungus showing arthroconidia with disjunctive cells. So, but some modes could be identified based on their arthroconidia characteristic, usually combined with other morphological characters. So I have discussed about Coccidioides and Malebranche. This Neoscatlidium, so we can identify Neoscatlidium based on their arthroconidia. Okay, this Neoscatlidium is significant fungus causing nail infection. Okay, it's non-dermatophyte fungus causing nail infection. Okay, maybe we can identify also trichosporon by their arthroconidia. Okay, so these are a very few fungus that we identify based on their arthroconidia. But if you see any fungus, some fungus with arthroconidia, I mean, very difficult to say what fungus is that, because these arthroconidia are formed in large number of fungal species. Okay. Then we move on to modes that produce conidia direct from hyphae. Okay. Okay. In some fungus, when the colony matured, the hyphae produce conidia directly from the hyphae. So like this is the hyphae, and this is the conidia. So emerge from the hyphae. So these are the what we call it macroconidia, emerge from the hyphae as well. This is microconidia, single cell, multicellular cell. Okay. Okay. I would like to discuss about dermatophyte. There are many fungus that produce micro and macroconidia directly from hyphae, but probably dermatophytes are very important fungus. that behave like this. Like, I mean, they produce micro and macro conidia. Okay. So the difficulty is to differentiate three species of Trichophyton. You know, in dermatophyte, we have three genera: Trichophyton, Mycosporum, and Epidermophyton. It's not difficult, very easy to differentiate these three genera. But we have three species of Trichophyton, and this probably constitute more than 70% of dermatophytes we isolate from clinical specimen. So, but I visited several labs. I found it's difficult to differentiate these three species. Okay, so let me give some tips on these problems. Okay, we can differentiate these three species by looking at their microconidia. Okay, don't jump into macroconidia. Many staff, lab staff, when they think they are looking at the dermatophyte, they want to describe the macroconidia first. Okay, but actually you describe the microconidia first, because if you look at the microconidia of Trichophyton tonsuren, you see it has various shapes of microconidia, variety of shapes. So variety of shapes, microconida alongside hyphae is characteristics of Trichophyton tonsurans. In Trichophyton mentacrophyte, you see the microconidia are in cluster, or what we call it like bunch of grapes. Okay, and the shape is somewhat oval, whereas in Trichophyton rubrum, the microcondia are teardrop shaped and form alongside hyphae. So by looking at the microconidia, we can differentiate Trichophyton rubrum, Trichophyton mentacrophyte, and Trichophyton tonsurans. But if you see macroconidia, then it is a bonus actually. Because we can confirm the identifications of the dermatophyte. If you see various shapes of macroconidia, then the culture could be identified as Trichophyton tonsurans. In Trichophyton mentacrophyte, the macroconidia are seagull shaped, whereas in Trichophyton rubrum, we describe this one as pencil shapes. Then we move on to another group of mold to study their key morphological characters. Okay, molds that produce spores enclosed in a sporangium. Okay, sporangium. So the hyphae produce a specialized hyphae. We call it conidiophore. Okay, so this conidiophore swell at the tips, and this swelling part is called columella, and this columella is enclosed in a wall. We call it a sporangium, and spores are formed inside the sporangium. So this is the basic structure of a group of fungus in the Mucorales groups. Okay. Okay. If you look here, Mucor, Rhizopus, Rhizomycor, Lichtemia, Succinia are 5 genera of Mucorales, and they share the same morphological character. Okay, as we described in the previous slide. Okay, so they looks very similar. So how do we differentiate them? So we have to look for some key morphological characters to differentiate Mucor from Rhizopus, from Lichtemia, from Succinia. Okay. Okay. First of all, we can look at the difference in the sporangium shape. Okay, sporangium shapes. If you look at the Saxenia, it has bars of fly shapes. So it's the only fungus that has these sporangium shapes. Okay, so in Rhizopus mycorrhizomico, the sporangium are normally spherical or globus. In Leishthemia, it's piriform or teardrop shapes. Okay, then another character in mucormycete or mucorales are the presence of apophysis. Okay, apophysis is additional structure just before the columella, and it's not covered by the sporangium. So Leishthemia has apophysis in a conical shape. Apophosomyces has apophysis in bell shapes. Mucor has no apophysis. Rhizopus has very small apophysis. Then another character that we can use to differentiate mucorales is the branching of the sporangiophore. So in Rhizopus, there is no branching of sporangiophore, but mucor, Leishthemia, you can see branching of. This is the hyphae actually. This is the conidiophore, and you can see branching of conidiophore. Okay, this is non-branch. These are branch conidiophore. Okay, and lastly, another key morphological characters in mucorales is the presence and position of rhizoid. Rhizoid is root-like structure on the hyphae. Okay, mucor has no rhizoid. Rhizopus has rhizoid near to its sporangiophore. So we call this position of rhizoid as node. Whereas in Leishthemia, so the rhizoid is in between sporangiophore. Okay. So what's the problem with identifying mucorales? The problem is fungus like mucor, Rhizopus, it has very long sporangiophore. That's very long sporangiophore. So maybe if you look Rhizopus under microscope, you will see only this part, maybe this lower part. So to study and identify mucorales, we have to study the fungus part by part. I mean, you look for apophysis, you look for rhizoid, and also the branching. Okay, and probably you can use a table like this to identify the mucorales. Okay. Another problem with identifying mucorales is to look for the rhizoid. Okay, rhizoid is a structure inside the agar growth medium. They are not on the aerial part of the hyphae. So most lab in Malaysia, they use sellotape mounting technique to prepare microscope slide for fungal identification. So if you use that method, you won't get, very difficult to get the rhizoid. Okay, so you probably missed it, and you will never identify Rhizopus, for example. Okay, so the tip is the presence of rhizoid is best observed from teeth mount. So instead of using sellotape mount, you should learn and use teeth mount. techniques, okay? Because in this mound technique, we will dig a little bit of the growth in the agar, so there will be more chances to dig out a rhizoid. Or if you culture the mold in slant, so I'll still culture the fungus in slant, tube slant. They are not using a plate actually. So you can look for the rhizoid using dissecting microscope on the back of the agar slant. Okay. Okay, we move on to mold that produce multicellular conidia, what we call it macroconidia. So this is probably a more interesting fungus to identify, give you less headache, okay? Because you can identify the mold by describing the macroconidia, okay? So this is macroconidia because it has many cells, okay? So these are some example of fungus with macroconidia. So this is microsporum. There are 1, 2, 3, 4, 5 cells here. Okay. This is Cavalaria. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. Mostly 4 cells in the macroconidia. This is histoplasma, even the 1 cell, but the size is so big. It's so different from the microconidia it produces. So we still call this one macroconidia. Okay. How do you identify fungus based on the macroconidia? Okay. You should actually learn how to describe macroconidia. Actually, when you see under microscope, you see macroconidia, you can jump because now you are very near to identification of the mold, but you must learn how to describe the conidia to confirm your identification. So to describe macroconidia, first of all, you have to count actually the septum or septa inside the conidia. Conidia are cell, okay? So it can be a single cell or multi-cell. So if you have one septum, you have 2 cells inside the conidia. Okay, a conidia without septum is called amiroconidia. You probably will read this one in fungal atlas, okay? Because fungal atlas always describe conidia, whether micro or macroconidia. There will be a section in all mycology atlas describing fungal conidia. If it has 1 septum, so the conidia has 2 cells, this is called the dimoconidia. If you have more than 2, there are 2 types of macroconidia in this group. One with it has only transverse septa, multi-cell, more than 2, but only transverse septa. So this is called pragmoconidia. Okay, and in other mode, it has transverse as well as longitudinal. Transverse, this is longitudinal, multi-cell, but has transverse septum and longitudinal septum. So this amiroconidia, didymoconidia, pragmoconidia, dictyoconidia are actually some of the key morphological characters found in macroconidia descriptions. Okay, so from subtension, next we have to describe shapes of the conidia. So these are the compilations of shapes of conidia of fungus that you can isolate from clinical specimen. So if you describe the conidia, it's probably fall into one of these shapes. But on this side, I have listed the more common shapes of fungal conidia. Okay. Then look at the conidial surface. Conidial surface, okay. This is whether you can see anything on it. In this epidemophyton, there's nothing on the surface. We call this one smooth. Smooth. Whereas in these three groups, three species of fungus, there are some kind of projections on the conidial surface. So you can actually broadly call this conidia as rough. Rough. If you don't know how to describe the roughness, you can actually call rough surface conidia. Okay. Okay, and then if you have a micrometer in your microscope, you should determine the size of the conidia. It's probably help you in species determination, and also color of the conidia, whether hyaline or dermatiaceous. Okay. Okay, then we move on to molds that produce single cell or unicellular conidia. Actually, from my experience, more than 60% of the time, you will encounter these groups of mold from clinical specimen. Okay, so but because it is single cell conidia, so it's very difficult to identify these fungus based on the conidial characteristic. Okay, because no septum, and maybe you can see roughness on the surface, or the color is hyaline or black, but that's all. And these three characters are shared by hundreds of molds. Okay, so how do we go about identifying fungus that produce single cell conidia? So what are the key morphological characters in this group of fungus? Okay. Okay. This single cell conidia. Okay. First of all, if you look at single cell conidia in your slide, you should determine whether the conidia forms sticky mass after being released from conidia forming cell. Okay, okay, this is what we call it sticky mass or balls of conidia. Okay, this characteristic is very important. Okay, because some fungus, like in this Pylophora, okay, Pylophora is characterized by formations of balls of conidia at the tips of the pylites here. This is acromonium. This is the pylites, and you can see balls of conidia. Of course, it can fall down because I mean it's just a sticky mass. I mean they just aggregate there. There's nothing actually hold them, so it can fall down the pylites. Okay, but at the beginning they form balls of conidia. Okay, and then in other fungus, the conidia form will be in chain, okay, in chain. Okay, instead of balls of conidia here, they are in chain. Okay, so you look at these two interesting mold. One is Murnihayati, the other one is Tachybotrys. Okay, they have very similar conidia forming cell. This is the conidia forming cell, very similar. Okay, but in Tachybotrys, the conidia are formed in sticky mass, whereas in Murnihayati, they are in chain. See, because of chain, the names of the genera change also. So it's very important to confirm whether your fungus produce chain of conidia or sticky mass conidia. So two very important key morphological characters. And if the conidia are in chain, there are two types of chain. Okay, there are two types of chain. Luckily, there are two, not five, not six. Okay, so one is we call it catenate. We call chain of conidia catenate if there are more than 3 conidia per chain. If up to 3, we call it catenulate. Okay, and if the chain of conidia, if you look at this chain of conidia, you look at here, the conidia is smaller than here. So this is youngest conidia. This is the oldest conidia. So the youngest conidia is at the beginning of the chain or at the apex of this conidia forming cell. So this chain, when new conidia form at the base of the chain, we call this chain basipetal chain. Okay, basipetal chain. Okay. So these are example of fungus with basipetal chain. So I'm sorry, this one should be sorry, this is another chain. We have basipetal. Here we have a catenate, but acropetal chain. So if you look at this chain of conidia, this conidia at the base here, at the beginning of the chain, bigger, that means older. This is the youngest conidia in the chain. So see, this is different from the basipetal chain. So this chain is called acropetal chain, can be seen in Alternaria. So this one should be acropetal chain as well. Okay, and also in Predosporium. Okay. Okay, we finish with conidia in chain. The problem with conidia in chain is that when you prepare a microscope slide to study the fungus, so this conidia, they are fragile actually. So this conidia will detach from this conidia, and you probably see conidia not in chain at all. Okay, so that is one problem in the determining of types of chain in fungus, types of conidia chain. Okay, there are fungus which form single cell conidia but solitary, not in chain. Okay, not in chain. And they are attached to the hyphae or conidia forming cell. Okay, these are examples of fungus that produce solitary and sessile. Sessile means directly attached to the hyphae, like in dermatophytes, Keratophyton, Scedosporium, auranticum, and Spotric shankai. You know, in Spotric shankai, they produce daisy flower conidia at the tips of a special hyphae, but there are also conidia formed alongside hyphae. So this conidia are solitary and sessile. Okay? But in some fungus, if in some Mohd, if you look at the Mohd, you probably see that the conidia are not directly attached to the hyphae, but the hyphae produce a short, small, tiny stalk. We call it denticles. So very important terms, denticle, very important key morphological character. We call it denticle. So the conidia sit on the denticle, okay? So you have to confirm whether the conidia that you see are sessile or they are on denticles. Maybe in this case, in this situation, you need to use all immersion. So most of us think that when you work in mycology lab, you don't need the all emission. Actually, you need the all emission 100 magnification lens very much actually in mycology, okay? Because there are very, very small key morphological characters that you need to use all emission oil to identify the structure, to locate the structure. So even in Sportric Shankai, okay, you look at the flower, actually these conidia are attached to this part is called conidiogenous cells or conidiophore through denticle actually. That's why it looks like daisy flower, okay? Okay, we finished about conidia, microconidia on denticle or sessile, whether they are in chain or in a sticky mass. So then we should look for where actually this conidia, single cell conidia are formed, okay? Because if you see macroconidia, single cell, or even macroconidia, there must be cells that produce it, okay? Not all conidia, micro, come from hyphae, direct from hyphae. So in many fungus, there are specialized cells that produce the conidia. In this example, Sportric Shankai, this is the hyphae, this is the hyphae, and this hyphae produce a specialized hyphae. We call this conidiophore. Conidiophore, and conidiophore produce the microconidia. So in this case, it produced at the tips or near end of the conidiophore. Okay, so conidiophore is very important conidiogenous cell, but the problem, they maybe look similar to hyphae. So if you prepare the slide badly and this macroconidia separated from the conidiophore, you may think this is hyphae, branch of hyphae, and you see scattered microconidia, and you have no idea where this microconidia come from. Okay, so anyway, this is important key morphological characters in mold. We call it conidiophore. Okay, two more example of fungus with conidiophore is one is Blastomyces dermatitidis. So this is conidiophore, which looks similar, almost similar to hyphae, and it produces single cell conidia at the tips of the conidiophore. In Fusarium, this is the conidiophore. It produces conidia in balls from the conidiophore. Okay, another terms key morphological characters in fungus that produce conidia from conidiophore is the what we call it sympodial arrangement of the conidia. Actually, sympodial is a conidiogenous cell growth which results in the development of conidia on a geniculate or zigzag rachis. So we call this part geniculate or zigzag rachis. Normally, this morphology is seen at the end of the conidiophore. Okay, geniculate, zigzag rachis, or we call it sympodial. This is very big term actually in mold identification. Okay, sympodial conidiophore. Okay. We can see this sympodial conidiophore here also in Bouveria species. This is the conidiophore. See how the tips of the conidiophore elongated and it formed conidia, but you can see here it's in the form of zigzag rachis, or we call it geniculate. So this is sympodial This is what we call it geniculate conidiophore. Okay, and these are three more examples of geniculate conidiophore. See, this is the hype. This is the conidiophore. This is still in a conidiophore, but this part geniculate. This one straight. Okay, in bipolar too. Okay, someone posted this one in Twitter. I actually don't use Twitter. I don't know why, but I found this one in Twitter. Someone posted this one. Conidia seen on rachis, see? Rachis. So this is the rachis. Okay. So Mohd identify Acrodontium salmonium. So it's very important actually to learn key morphological characters, and conidiophore in rachis form is one of the important key morphological characters. Okay, now we move on to another conidiogenous cell. We just finished with conidiophore. Another important conidiogenous cells is, we call it phyllite. Phyllite, okay? Phyllite. So this is the hype. This is the phyllates, okay? This is hype. This is phyllite. So this phyllite in Acromodium, the phyllite produce the conidia from inside the phyllite at the tips of it, and these conidia remain in sticky mass. So this is in Pylophora. So phyllates produce conidia, and the conidia arrange in sticky mass after release from the phyllates. So another terms related to phyllite is whether at the tips of phyllite you can see very clear what we call it collarette, structure called collarette, like a collar. So this term comes from collar, collarettes. If you can see it, that means the collarette is conspicuous or distinct. Whereas in Acremonium, the collarette is indistinct. Okay, so if you look at phyllates, you should also look at the tips of the phyllite to determine whether the phyllite has distinct or indistinct collarette. So the collarette. Okay. Another features of phyllite is whether the phyllite has a basal septum like this or no basal septum, like this, and like this. So phyllite without basal septum is called adelophyllates. Another important terms, characters, morphological characters in molds, adelophyllite. Because some mold produce only adelophyllates, no discrete phyllite. Whereas in this Paleomoniopsis, it produces both adelophyllite and discrete phyllates. Okay. Okay, we know phyllite produce conidia, whether in balls or in chain, and whether it has collarette or not. But another features of phyllite is that it probably comes in various shapes. Some phyllates of molds are very unique. Like in Pylophora, the phyllite is described as vase shapes, like vase, flower vase. It's very beautiful. Flower vase with balls of conidia. So this Pylophora verrucosa. And in Acremonium, you can see here, very long phyllates, but tapering towards the end. So this is called owl shapes. Okay. In Trichoderma, you can see bend, irregular bend at the tips of the, in this case it's annelide actually, not phyllite. Okay. And in Exophylla moniliae, the phyllite is beaded. Beaded looks, mean the structure we call it beaded structure of phylates. Okay, so there are a variety of shapes of phylates. So atlas, mycological atlas, will always describe the shapes of the phylates. So when you look at phylates, think about collarettes, think about basal septum, and think about the shapes of the phylates. There's another corneal cell which look similar to phylates, function similar to phylates, that is produce cornea, either in chain or in balls. But we call these corneal cell as analytes, and it's not easy to differentiate analytes from phylates. So if the phylates is like this one, vase shapes or flash shapes, then probably you can look carefully at the tips of this corneal cell. If you can see extended area there, this is probably analytes, because in analytes, the length of analyte increases after every production of microcornea. So it keep on elongating. Okay, whereas in phylates, the length of the phylates remain the same no matter how many cornea it produces. Okay, if you have a good microscope that can enlarge this cellular morphology of mold under microscope, then it's probably easier for you to see the analytes, and this elongated part is called annulation. Okay. So this is in Schizosporium prolificans. Okay, these phylates can be directly from the hyphae, or in some fungus, they are on conidiophore. So these three examples, this is simple conidiophore, that means unbranched conidiophore, and at the tips of conidiophore are phylates. So phylates are supported by conidiophore. So the phylate produces conidia actually. So in some fungus, you see the conidia cell like that, not phylates alone, but phylate with conidiophore. Okay. In some fungus, the conidiophore branch is not simple conidiophore, even though at the tips of conidiophore, it produces the phylates or analytes, but the conidiophore branched. So there are two types of branching in fungus. The most common one is penicillus or penicillate types of branching. Okay, the conidiophore branch giving a brush-like appearance like this. It branch and it gives brush-like appearance. So this is penicillate types of branching or penicillus. That's why this fungus is called penicillin. Another types of branching is called verticillate types. Okay, branches arranged in verticils or whorls. Okay, or you can see the branching is at the same point. So same one point, you have two branch of conidiophore. Here you have three conidiophore from the same point of the conidiophore. Okay, so this is called, we call it verticillate types of branching. Okay, so if you see branching of conidiophore, you should also determine whether it is verticillate or penicillate. Okay. Okay. We come to the last groups of mold before we discuss about some tips in learning or in determining the key morphological characters. So in this fungus, it produces conidia in fruiting bodies such as pycnidia. Maybe some of you have this experience. They isolate one mold, prepare the microscope slide, and look at the microscope, but they see hyphae and this big structure. We call this one fruiting bodies. Okay, so inside the fruiting body are the conidia. So one types of protein body is called pycnidia. Pycnidia. Okay, pycnidia has opening. We call it ostiole. Inside conidia are the hyphae and pileates. So these pileates produce conidia, and this conidia will be released when the pycnidia mature. Okay, pycnidia mature. So if you see fruit bodies, very big structure compared to the hyphae here, and usually dark color, and you see a lot of single cell conidia release from the structure. So this fungus could be called oomycete. Okay. So sorry, it's called coelomycete. Coelomycete. Okay. So these are some common coelomycete that we can isolate from clinical specimen. For a more complete list, I have here, not complete actually, but we have many more coelomycete that you can isolate from clinical specimen. But it's not easy to identify the coelomycete because you have to describe the conidia, you have to describe the spilite inside here, and it is not easy to crush this structure to study pileates and conidia. Okay, so you need a special training on this. But if you can see conidia from pycnidia like this, maybe you can report the isolate as coelomycete. Okay, coelomycete. Okay, so come to the last part of the lecture or presentation. Unless distinctive spore types are formed, identification may not be possible. So if you want to identify mold by cellular morphology, it's not possible if you don't see spore. Okay, if spore is not formed, it's not possible to identify mold to genera or species level. Okay, maybe you can say stomatiaceous mold only, it's basidiomycete, so up to this level only. But if you want to identify mold to genus, the species level, you must see the spore. But the problem is many clinical isolate do not sporulate on separate dextrosega. So I think most, maybe all labs in Malaysia use separate dextrosega as their primary culture medium for isolations of mold from clinical specimen. Okay, but the problem is many mold do not sporulate on SAP, but not all, but many. Okay, so yes, we all know that conidia are formed when the colony is matured, but how do you know colony has matured or not? Okay, so because it may take several days, it may take more than a week, it may take 10 days for the colony to get matured and produce conidia. So if we want to study mold for their conidia, okay, especially on medium like SAP, so the best technique to use is needle tease or squash mount technique. I have visited many labs in Malaysia, and they don't have this technique. It's actually a very cheap technique. What you need to have is two needle, stiff needle, okay? And you sterilize the needle and pick the colony, a portion of the colony, and look at the microscope. That is needle or teeth, needle teeth or squash mount technique. So use sterile technique, remove a small portion of the colony with inoculation needle and mount it in drops of lactophenol cotton blue on a clean microscope slide. So the portion of the colony may be teased using two needles, or otherwise cover with it with a microscope cover slip. Squash the preparation with the butt of the inoculation needle. I have actually a video of this, but I don't know whether it will play in this presentation or not. If you want to see the video, I will show it after my presentation. Okay. Let's move on to this slide. Okay. In a teeth mount or squash mount, you have to pick mycelium from 3 different points in order to make sure that the fungus not yet producing spore. Okay, so look at this colony. If you look at this colony, because fungus grow outward. Okay, so Z is where the hyphae is still very young. So X a point where the hyphae already is older, and probably it's already forming spore here. Okay, so you should pick fungal mycelium for teeth mount preparation from 3 points of the colony. Okay, so many labs, if they do teeth mount or tip mount, they just simply pick anywhere in the colony. So, but actually, if you use tip mount, it's better to have the tip from this X to Z, a little bit longer tip. So if the colony is not too big, or otherwise you need 3 tips, one here, and one there. Okay, so this is very important tips if you want to make sure the fungus has sporulate or not. Okay. Okay, another.
Dr. Fuat, sorry to interrupt. Okay, we may have 5 to 10 minutes left for the presentation. Okay.
Well, I will expedite my talk. Okay, now probably you need sporulation media. You must have sporulation media. Okay, every lab must have sporulation media. That means when they grow their fungus on the primary isolation media, like said, they must subculture it on media other than SAT, media that induce sporulation. Most lab will use PDA, potato dexosaga, while some lab use other medium. So, and also you should learn about slide culture technique. Okay, slide culture technique. Slide culture technique also will enhance inducement of sporulation. Okay, but it's a bit laborious like this. Okay. So we need to see the required essential identification characters. Not only we must be able to see the conidia, but also how the conidia arrange, where the conidia come from, and types of conidia forming cells. Okay, so most lab in Malaysia use cellular mount techniques. And if they use a tip mount, they can make a better teeth mount, cellular tip flight mount by pressing the tips on the colony. Not only touch the colony, but press the tip onto the colony. So that will give you a better chance to collect hyphae, not only conidia. Okay. And probably slide culture technique is the best technique to study key morphological characters, because you don't disturb the conidia, the hyphae, the pileates. So everything are fully intact. Okay, lastly, this is my last slide. I know it's not easy uh, to look for these key morphological characters. You don't even memorize the terms used. So you have to get familiarize yourself with the key morphological characters. Okay. Today we learn about mycology terminologies. Okay. So what you should do is to learn further these morphologies by going for, let's say, attachment to the lab that keeps fungal culture and slides such as at MR. So maybe you can try identification quiz and websites such as Mycology Online. It's a good way to learn key morphological characters. And you should do identification in the lab continuously and study the isolate after receiving. Many labs send their culture for identification to the reference lab. So when they receive the report, they should come back to the culture and study the isolate by using reference atlas. Okay, this is very, very good way to learn key morphological characters. And also you should look for photos in website, and also you can study photos in atlas. So that's all what I want to discuss about identification of Mohd by cellular morphology, and some suggestion or tips for you to get to be better and better in doing fungal identification from cellular morphology. So thank you very much.
Thank you very much, Dr. Fuat, for the presentation. So we are now in the question and answer session. So I'd like to open the floor for any burning questions. You may raise hand and you will be unmute to directly ask question to our speaker, or alternatively, you may also just drop questions in the chat box. Okay. I think they are all mesmerized with and overwhelmed with all the knowledge shared by Dr. Fuat. So while waiting for others to ask question, maybe I can start with a question to Dr. Fuat first. Okay. Okay, sure. For example, Dr. Fuat, this is just a mere example to illustrate our difficulty in morphological identification of mold. For example, we have a mold and we able to do the LPCB slide, and then when we examine for the morphology, for example, it looks like Phaeociliomyces, but the colony characteristic does not match the description. So where we should go from that, Dr. Fuat?
Okay, thank you for the question. I did not I did not discuss the colony morphology in more detail because you see, colony morphology is affected by the media you use, affected by many factors. So the colony morphology colors may change. Even the age of the culture will change the colony morphology. So it's very difficult to start your identification from colony morphology. Actually, you should start from the cellular morphology and then try to confirm the identification by looking at the colony. But, you know, this colony, you must make sure that you use the same culture media. And you must check that some fungus has a range of colors, some range of morphology of colonies. Okay, so if you are very sure by cellular morphology the fungus is bacillomyces, but the colony, according to what information you have, is not fit to bacillomyces, then you should stick to your cellular morphology. Okay. Yeah, stick to your cellular morphology. That's why it's very, very important for you to study the key morphological characteristic, because that structure don't change, but colony color or characteristic may change by various factors.
Okay, thank you so much. This is very well answered, Dr. Fuat. We just have few comments from our participants. Dr. Humaira was very mesmerized with everything shared during this presentation session. And another participant, Intan Sofia Alias, acknowledge that this CME is very informative. Thank you so much. So she asked regarding for the slide presentation. So this we realize first with Dr. Fuat whether Dr. Fuat we change first into PDF form whatsoever. Later we will inform to our participants together with the recording. So please just have a patient first before we do all the needful and sharing the links to you later. Okay. All right. Dr. Fuat, I think they all thank you very much.
Dr. Muni, sorry,
Yes.
Dr. Muni, there's 2 questions in the Q&A box.
2 questions. Hold on. Okay, sorry, I missed that. Okay, from probably Dr. Lee Pei Chen, our participant asked how to troubleshoot non-sporulating fungal from sterile samples. Okay, Dr. Fuat.
Can you repeat the question?
Okay, the participant asked how to troubleshoot identification of non-sporulating fungal from sterile samples. So they got the sterile samples, they already have a growth, but the isolate when they examine is a non-sporulating fungi. So I guess the clinician is they are worried this non-sporulating might be one of those significant isolate. So how do they go about with this non-sporulating fungal?
You must first of all make sure the fungus do not sporulate, even if after culturing it on potato dextrosega. But if still non-sporulating, you are not able to identify the fungus, then you have to go for identification by DNA. Or if you have Multitoph, you can identify it by using Multitoph. Okay, and also to determine whether the fungus is significant or not. Actually, you should look at the the clinical specimen directly. So we call it direct microscopic observation. If you see a fungal element in your specimen, so what grow from the specimen probably significant. Okay, another way to decide whether the fungus is significant or not is by repeat sampling. You sample again from the patient whether you isolate the same non-sporulating fungus. Okay, and also you can culture the specimen by doing a spot inoculation and see whether all the spot grow with the same fungus. So that's how we determine the significance of the fungus growing from the specimen, even though it does not sporulate. But the easiest way is to send it to a lab, any lab that can do molecular identification.
Okay, thank you, Doctor, for the answer. We have another, probably the last question, if there's no more question after this. So this question is how to differentiate firelight and Cornidia 4. So this is part of the lecture just now. So maybe further explanation they seek from Dr. Fuat. So please, Dr. Fuat.
Okay. I expect this types of question because if you look at the phyllite, the conidia are always at the tips of the phyllates. Okay, they are always there because the conidia is produced inside the phyllite and released at the collarette, near the collarette there. Okay, so but if you see a conidia not at the tips of the conidigenous cell, it is not phyllates. It's probably conidiophore. In some fungus that produce conidiophore, you see conidia at the tips of the conidiophore, but maybe you can see the denticles at the tips of the conidiophore. So they are not actually coming from any phore at the tips of the conidigenous cell. So meaning that fungus with conidia, not at the tips of the conidigenous cell, possess conidiophore, but not all fungus that produce conidia at the tips possess conidiophore. It's probably phyllates or annelides. So morphologically, it's very difficult to differentiate conidiophore from phyllates unless if you study carefully at the tips of the conidiogenous cell. So whether you can see collarettes or not, or you can see denticles or not, or whether the conidia are at the tips, but actually they are not all at the tips. They probably some conidia produce a bit down of the tips. So maybe sessile, maybe on denticles. So that is conidiophore rather than phyllates. Okay, but my advice is don't worry about what you see conidiophores or phyllates. What you should do is to learn, let's say 50 most common fungus in clinical specimen, and you should remember by heart. For example, in Scoporopsis, the conidigenous cell is analyzed. In Aspergillus, phyllates. Okay, Schistosporium, analyzed. I remember that by heart actually. So don't try to look, when you look fungus under microscope, don't try to think, oh, this is phyllates or analytes or conidiophore. That will be a very difficult answer to a question to answer. Okay, don't ask yourself that, but just identify. I'm sure this is the conidigenous cell. That is very important. You must make sure that you can see conidiogenous cells rather than to determine whether it's phyllates or conidiophore. But there are some features that you can use to differentiate phyllates from conidiophore if you're lucky. Okay.
Okay, thank you. We have one attendee raise the hand. I just want to allow our participant Yokai Ji. Would you like to ask question to Dr. Fuat? We already unmute you, so you can ask anything if you wish. Okay. Okay, maybe just by mistake accidentally press the raise hand button. So I think that is the end of the question from our Q&A box. And also I would like to thank Dr. Fuat for all the shared knowledge and also your experience in the field of medical morphology, especially in the identification of mold by morphology. So this is definitely a knowledge that we won't be able to acquire within a short period of time. As mentioned by Dr. Fuat, it is a continuous process of learning. And I would like to share one Malay proverb that saying, "Jauhari juga yang mengenal manikam." So this is fungal morphology in a nutshell. So with increase in knowledge and more experience, we will be able to achieve a level where we can recognize the mold by its morphology, even though there's certain limit that we need to then refer to molecular techniques. Okay, with that, I thank you for all the participants. Today, maximum participants that we reached was 117. So very much thank you for everyone. And not to forget to claim your MMA CPD point by scanning the QR code. So with that, I thank you all very much until we see you in the next MMIG webinar. So have a pleasant day ahead and we see you later. Thank you and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.